Welcome to my channel Srinivasa Biology Tutorials. For this section to discuss class 11 Biomolecules part 4. Now you discuss about as any chemical reactions of enzymes. How do you understand the enzymes? Let us first understand the chemical reactions. The chemical compounds undergo the typical changes. Okay, chemical reactions are nothing but what? Here any enzymes to get participate the reaction to get produced as in a one structure to another structure. How is that possible? Because there some reactions will be going on. Here some reactions will be going on. All physical changes are simple to refer to change the shape of without the breaking of the bonds. Here they having as in one enzyme. Here they having as what? One enzyme. This enzyme they get bind with as in a particular substrate. You get bind with as in a one particular substrate. What happened? Their physical change will be the states will be they get not change. Okay, will the ice the melts the water, or when the water becomes as a vapor, these are completely the physical process. These are completely what? These are completely the physical process. Now, however, the bonds are broken and the new bonds to get formed during the transformation, this wild is called as in a chemical reactions. When they get bonds, what the carbon-carbon bonds is there here. So these bonds to get break and to get form as in the new forms. To get forms as what? The new forms or new the compound is formed, that is called as what? In a completely chemical reaction. For example, like given here, the barium hydroxide to get bind with as in a that H2SO4 to get formed as in a barium sulfate. To get formed as what? Barium sulfate like. Okay. Now, is in inorganic chemical reaction the similarly hydrolysis of the starch glucose into as in a the organic chemical reactions, the rate of the physical or chemical process refers to as an the amount of the product to formed as any per unit of time. So per unit of time also to get formed as a product can be there expressed as any the rate is equal to that rho p by rho t, the product divided by as any rate. Okay. Now the given as any rate can be also called as any velocity of the direction. Here simply the rate is equal to the rate also is called as what? Velocity of the in a direction of the specified, that is in a direction of the specified. Now rate of the physical and chemical process the influenced by as in temperature among their factors. So temperature, pressure like these factors to affected by as in that rate also. Okay. Now the general rules the terms is in a rate of the double or increase is of the every 10 degrees centigrade change there either direction. So every 10 degrees centigrade automatically the chemical reaction will be to get change. Now the catalyst reaction the process adds in that rate vastly the high constant the high than that as in uncatalyzed ones. So sometimes what happens, sometimes reaction also does not takes place. When the enzyme catalyst the reactions are observed, the rate would be there vastly high then the uncatalyzed reaction. For example, the carbon dioxide they get combined with as an oxygen to get form as what? The carbonic acid will be to get forms. Okay now, like so this is in a uncatalyzed reactions also to get forms. The presence of the enzyme here carbonic anhydrase is what? It is in a catalyst, nothing but as in a enzyme. Okay, that is. Now if in a case the absence of the enzyme, in here the reaction is very slow. In case catalyst is absent, what happened? Here the reaction is slow. Without any 200 molecules of the carbonic acid to form as in within 1 hour. After when using as an enzyme present, the cytoplasm is called carbonic anhydrase enzyme. The speed will be to get form as in the diagrammatically as in 6 lakhs molecule to form as in every second. So here 1 hour to get form as in 200 molecules. Here only 1 second to get forms as in how many molecules around as in they are 6 lakhs molecules. Okay now, like depends upon what? It is complete depends upon the what in a enzyme. So that completely the reaction about as in 10 million times the power of the enzyme the incredible in it. So this is the responsibility of the enzyme. Now there are the thousands of types of enzymes is catalyst the chemical the metabolic reactions. The number of enzymes are participate as in a metabolic process. The multiple steps of chemical reactions, when each step is catalyzed by as in the same enzyme, the complex the different enzymes is called as in a metabolic pathway. Example as in a glucose convert as in a glucose into convert as in 2 molecules of pyruvic acid. So one product how do they get formed? To get participate as in the number of the enzymes are get participate, then only to get form as in a particular compound. Okay. Now Actually, the metabolic pathway, the change of the glucose becomes as a pyruvic acid through as in a different enzymes catalyze metabolic reactions. So here glucose is converted as the glucose 6-phosphate, fructose 6-phosphate, fructose 1 gamma 6 phosphate, like the different catalysts that participated to get formation of two molecules of their pyruvic acid, to get formation of pyruvic acid. Okay, when you recall the study of the respiration plants, the 12 chapter 12 to study about as the reactions. So the respiration plants are given as in completely all reactions also. Now this stage of this should be known as this the metabolic pathway, or 1 or 2 the additional reactions give as any variety of metabolic end product. So here 1 or 2 their metabolic process also to get forming here. In this about the skeletal muscle under the anaerobic conditions, the lactic acid to form. So anaerobic conditions also here to get form as what? In a lactic acid. To get form as what? Lactic acid. Under the normal condition, aerobic condition, the pyruvic acid formed. In normal conditions, pyruvic acid, and anaerobic conditions to get form as in a lactic acid. Now you see that it's same that in yeast cell during the fermentation. Now see that in a yeast cell, the same process to get produced as what? Ethanol or alcohol. Ethanol, alcohol also they produce. Depends upon the conditions, different product will be they get produced. So depending upon the environmental factors and different enzymes to get forms as in a different compounds. Okay. Now to discuss about as in a the rate of now to discuss how do the enzymes bring about such high rate of chemical conversion. Understand we there should be study about the enzyme a little more. We already understand the idea of an active site. So already you know that about some information about as an active site. So this is an active site, right? It is an enzyme. In this enzyme, as in a pockets, it is called as what? Active site. It is called as what? Active site. Now the metabolic conversion refers to as a reaction, the chemical which has converted as a product which is called as a substrate. So one substrate to get bind with as in this part, this part is called as what? Substrate. The enzyme, the protein, they are three-dimensional structures, including as an active site, is converted as substrate in product. The symbol they given as in S and P. So here substrate is S, that is in S, and product we denote as in what? P. Product is what? P is now understood that the substrate S it is bind with at enzyme active site. Enzyme active site within the given the cleft or pockets. It is substrate to diffuse towards the active site. So for example, you take it here, it is in a enzyme. It is the enzyme, it is the active site. It is what? It is in a active site. So in this active site to get bind with as what? Substrate. Here bind with as what? Substrate. Now this complex formation translates the phenomenon during the state where the substrate to bind to the enzyme to active site, the new structure is called as a transitional state structure is formed. So here it is the enzyme and it is in its substrate. Yes, here when to get enzymes and substrate to get bind, here to get forms as what? Enzyme substrate complex will be to get formed. Here otherwise the structure the substrate to get transformed into as a structure to form as what? Final to get forms as a product. Final to get produced as what? Product. On this pathway of this transformation must to go through that is called as in a transitional state. It is must to is called as what? It is in a transitional state structure. Okay. Now transitional structure that production. See here it is in a completely when you see as a graph of the enter the formation of product. It is in a without enzyme. It is in a it is without enzyme. It is what? Without enzyme. It is what? With enzyme. It is with enzyme. Like okay. Now here there could be more there are altered structure state between the stable, the straight and the product. So finally we getting as what? In a substrate and to get bind with an enzyme active site to get producing as a product. There implicate in the statement the flag to all other intermediate structural states unstable. If the stability is something is related to energy in the status for the molecular structure. It when to get formation of the product, it is changed as in their molecular structure also. Here hence to get looked as in a they are pictorially through the in a graph look like as something as figure 9.4. So in the 9.4 to given as this is here, it is in a without enzyme, with enzyme. In this structure is called as what? This is in a transitional state. In the transitional state you get producing as a product. Okay, here the given explanation also, the y-axis represent as a potential energy contained. So here they given as an x and a y. So here it is in a y-axis, it is as in a potential energy, and then here this x-axis represent as in the to structural transformation state through the in a transitional state. So it is called as what? It is a transitional state. It is what? Transitional state. You would be notice the things, the energy is difference between that in a S and P. So here it is the substrate, here is P. It is they having two different energies as to get forming. Here P is lower level, yes, the reaction to exothermic reaction. So it is this type of reactions is called as what? These are in a exothermic reactions. Exothermic reactions. Now one need to the supply the energy by heating to orderly to the product. So automatically once the enzyme to get bind with as in a substrate to get forms as what? To get released as in that product. To releasing as what? It is in a product will be to get formed. Like now, however, where there exothermic, there spontaneously reaction on the endothermic or energy is releasing the reaction in the S to go through the much high energy state or transitional state. So in case exothermic reactions or endothermic reactions to compensate, they require as what? Energy is requiring for the reaction. Then what happened? To get releasing as in a productivity. To get releasing as what? Productivity is formed. Okay. Next, the difference in average the energy content is the formation of the transitional state is called as an activation energy. When to get formed in this energy is called as what? Activation energy. Enzymes eventually bring down to energy barrier to make that in a transitional S to P is more easily. So here energy move down to as an A to breaking as an A energy barriers in the transition state S to P. S to P. Now we discuss was nature of enzyme action. Now discuss nature of enzyme action. The each enzyme has the substrate. Here nature of the enzyme. Each enzyme E has in substrate to bind with the active site the molecule that highly the reactive enzyme the substrate complex. So here the enzyme here mostly here enzyme to get bind with as an A substrate to get form as in A enzyme plus substrate complex. Enzyme substrate complex is produced. This complex should short-lived that dissociated with as in a product. It is short-lived to get forms as what? To get forms as in a product. To get form as what? Product. And this unchanged the enzyme, the intermediate formation of enzyme product EP. So when to get form as what? Enzyme plus product. To get form as what? Enzyme plus product. See the E plus S to get form as an enzyme substrate complex, and EP to get forms with an immediate to get form as what? Enzyme plus product will be they get separate. This catalyst, the cyclic, the enzyme action can be there described with the following steps. So in this total process, how do they going? They given as an A one step by step. Now say that first in that first step here the first the substrate to bind with as an active site of the enzyme, the fitting of in active site. So in that active site the substrate to get binding here, to get binding. And binding the substrate, they include the enzymes to alter the shape, the fitting the more tightly around the substrate. So different enzyme substrates as present. The different substrates to get bind with as in tightly under the substrate conditions. Now the active site the enzyme, now the close the proximally the substrate to break the chemical bonds substrate and new enzymes product will be complex will be formed. So when to get form as in a product, what happened? This product will be to get separated, P will be separated, and then here again the enzyme to get bind with as an A another substrate, to bind with as an A another substrate, and then the enzyme they release it to product, the enzyme to get released as a product, and the reaction and their free enzyme related bind with as another molecule, the substrate will be through as a catalytic cycle once again. So I say that one only. Here once to get product will be formed, what happened? Here enzyme also will be separated and product also will be separated. This enzyme to again to get go to and to bind with as an A another substrate. Okay, this is the incomplete nature of the enzyme. Now here factors affecting by enzyme activity. Now to discuss here the factors affecting enzyme activity. Now, the activity of the enzyme can be affected by as in change of the factor conditions can be altered their tertiary structure, the protein. The protein structure mostly they are affected by as in their complete enzyme action. The including as what temperature, pressure means the pH, and the change of substrate concentration to bind with as in the chemicals to regulating by its activities. So these are completely they are affected by as in their enzyme activity. Now once discuss one by one. The first one is here temperature and pH. Now to discuss what? Temperature and pH. See, in this temperature and pH, the enzyme generally their function is a narrow range of temperature and pH. Here they having as a narrow range of pH will be there present. See here they given graph also the pH and temperature. They given as a narrow temperature. They given as in a completely in a narrow temperature is given here. Now each enzyme show as in highest activity, the particularly the temperature and pH is called as an optimum temperatures. When they get followed by as in the constant temperature, the temperature is called as what? Optimum temperature and optimum pH. Now the activity there declines the both below and above optimums, both below and above optimums values. The lower temperature, the presence as an enzyme, they are temporarily the inactive. There were as in a high temperatures that destroy the enzyme activity because the proteins are denatured in the heat. So the temperature will be there decrease, automatically the enzyme activity will be also they get decrease. Like the concentration of substrate, they given as in a concentration of substrate. Here the increase of the substrate concentration, the velocity of the enzymatic reaction, the raise the first. So enzyme activity reaction will be the raise, they are first. The enzyme, the reaction ultimately reaches as a maximum velocity, that is Vmax. So to get reach as any that maximum velocity, that is called as what? Vmax, which is not expressed as in there any there further the concentration of the substrates. So it is not depends upon the another concentration substrates, because this is because their enzyme molecules they are few than the in substrate molecules. In faster saturation is molecules takes place. So here most of the saturation takes place on that in a substrate molecule. There are no free enzyme molecules to bind with as a additional substrate molecules also. The free enzymes to get bind with as an additional substrate molecules. Now here the activity of the enzyme also the sensitive, the presence of the specific chemicals to bind with as an enzyme. So particular the already enzymes also they mostly specifically they bind with as an sensitive. For example here the different enzymes to get bind with as, for example here they having this type of one substrate, this is also one substrate. So this bind only here to get enzyme activity will be there more, but in this substrate to get bind here what happened, this active site is very different, this substrate is different. So then what happened, inhibition is takes place. Now when the inhibitors closely that resembles the substrate, the molecule structure and inhibits the activity the enzyme, this is known as a competitive inhibition. So here mostly the different substrate, the same structure of the substrate to get bind another sides, what happened, the enzyme inhibition takes place. That is called as what? That is completely called as a competitive inhibition. Next, due to is the closed structure, the similarly within the substrate, inhibition is competes to substrate to bind with as an enzyme activity. So the enzyme here, this enzyme activity side, the different enzymes to get bind here, what happened, this is the RL substrate, these are the different substrate to get bind, the enzyme inhibition takes place. Consequently the substrate cannot be bind with as a result, the enzyme action is declined. So another substrate to get bind with as another substrate in the enzyme activity, what happened, the total the enzyme action will be there stop. Example they given here, inhibition of succinic dehydrogenase by the melanonic acid, or that is a melanoate, which is closely resembles structures. The succinate it is in that structure. So they are structurally the same. So hence here what happened, one place to get bind with another substrate, the inhibition takes place. Such competitive inhibitors often used in the control of the bacterial pathogens. This type of inhibition we are mostly used as they are controlling the bacterial pathogens like. Okay, this is in completely the enzyme action. Okay, the remaining to discuss in next class. Thank you for watching.